Welcome to today's lecture on molecular signaling pathways in biology. In this session, we will explore the intricate details of 12 important signaling pathways that play fundamental roles in various cellular processes. By understanding the mechanisms governing these pathways, medical students will gain insights into the molecular basis of diseases and potential therapeutic targets. So, let us dive into the fascinating world of signal transduction. In GPCR signaling, transmembrane GPCRs transmit signals from external molecules to inside the cell. GPCR activation leads to the separation of G proteins, triggering downstream effectors like adenyl cyclase or phospholipase C. Second messengers like CAMP or IP3 are generated, producing specific cellular responses. This pathway controls functions like neurotransmission, hormone secretion, and immune response. The RAS-MOPK signaling cascade plays a crucial role in cell growth, differentiation, and survival. When activated, receptor tyrosine kinases or GPCRs recruit and activate RAS proteins, initiating a phosphorylation cascade involving RAF, MEK, and MOPKs such as ERK1/2. These activated MOPKs then regulate gene expression in various cellular processes. Dysregulation of this pathway is linked to diseases like cancer and developmental disorders. The phosphatidylinositol 3 kinase, PIEK, signaling pathway is crucial for regulating cell growth, proliferation, and metabolism. Peaks phosphorylate the inositol phospholipid phosphatidylinositol 4,5 bisphosphate, PIP2, to generate phosphatidylinositol 3,4,5 trisphosphate, PIP3. PIP3 recruits and activates downstream effectors, including AKT, also known as protein kinase B, which triggers a signaling cascade promoting cell survival and growth. Dysregulation of this pathway is associated with various diseases, such as cancer, diabetes, and autoimmune disorders. The WNT signaling pathway plays a crucial role in development and maintenance of tissues and stem cells. WNT proteins bind to frizzled receptors and LRP5/6, leading to stabilization and nuclear translocation of beta-catenin. Beta-catenin then interacts with TCF/LEF to activate important genes. Dysregulation of this pathway can contribute to diseases like cancer and developmental disorders. The Notch signaling pathway is a highly conserved pathway involved in cell fate determination, differentiation, and tissue development. Notch receptors interact with ligands expressed on adjacent cells, triggering proteolytic cleavage and release of the Notch intracellular domain, NICD. NIC translocates to the nucleus and complex with transcription factors, activating downstream target genes. Dysregulation of Notch signaling has been linked to multiple disorders, including cancer, cardiovascular diseases, and neurodevelopmental disorders. The TGF-β signaling pathway is crucial for controlling cell growth, differentiation, and immune response. TGF-β ligand binds to specific receptors, activating SMAD proteins that regulate gene expression. Dysregulation of this pathway is linked to diseases like cancer and autoimmune disorders. The hedgehog, HH, signaling pathway plays a crucial role in development, stem cell regulation, and tissue maintenance. When HH ligands bind to PTCH, they activate SMO, leading to the activation of GLI transcription factors and the regulation of target genes. Abnormal HH signaling is associated with developmental disorders and cancers like basal cell carcinoma and medulloblastoma. The Janus kinase signal transducer and activator of transcription, JAK-STAT, signaling pathway plays a critical role in cytokine-mediated cellular responses, including immune regulation, hematopoiesis, and inflammation. Cytokines bind to their cognate receptors, leading to JAK activation and phosphorylation of STAT transcription factors. Phosphorylated STATs form dimers and translocate to the nucleus, where they modulate gene expression. Dysregulation of this pathway is associated with various diseases, including autoimmune disorders, leukemia, and lymphomas. The NF-kappa B signaling pathway controls immune response, inflammation, and cell survival. Activation occurs when inhibitory proteins are degraded, allowing NF-kappa B dimers to move to the nucleus. There, NF-kappa B regulates genes involved in immune responses, cell survival, and is associated with diseases like cancer. The Hippo signaling pathway is an evolutionarily conserved pathway that controls organ size, tissue regeneration, and stem cell maintenance. Central to this pathway is the MST1/2 kinases, which phosphorylate and activate the LATS1/2 kinases. Active LATS1/2 kinases phosphorylate the transcriptional coactivators YAP and TAZ, leading to their cytoplasmic sequestration and degradation. Dysregulation of the Hippo pathway has been linked to various diseases, including cancer and developmental disorders. The mammalian target of rapamycin, mTOR, signaling pathway regulates cell growth, metabolism, and autophagy in response to various cues, such as nutrient availability and growth factors. mTOR exists in two complexes, mTORC1 and mTORC2, both regulating distinct cellular processes. Activation of mTORC1 promotes protein synthesis and cell growth, while mTORC2 modulates cell survival and cytoskeletal organization. Dysregulation of this pathway is implicated in several diseases, including cancer, metabolic disorders, and neurodegenerative diseases. The CAMP-PKA signaling pathway controls many aspects of cell behavior, like metabolism and gene expression. It starts with receptor activation and leads to increased CAMP levels. CAMP then activates PKA, which phosphorylates multiple targets, regulating cell responses. Abnormal pathway activity is linked to diseases. In conclusion, comprehending the complexities of molecular signaling pathways is vital for understanding disease mechanisms and developing targeted therapies. We have explored 12 essential pathways, each with a significant role in cellular processes and disease. By studying these intricate details, medical students can gain insights into therapeutic opportunities. Let us continue our journey into the mysteries of molecular biology and medicine. Thank you for your attention, and see you in the next lecture.